from is there from our which are good friends and are experts in their field. Welcome to the delegates. Dr. Rama has planned, the team has planned an excellent scientific session. So welcome to you all.
Good morning, everyone. Respected Dr. Katos sir, other dignitaries. We welcome you all to this second RAJMICROCON conference, which is in the series of the Microcon, which is a very young association of ours. So I welcome all of you, and wish this conference will be a great success. Thank you.
Every scientific journey starts with an idea and discussion. Students are the backbone of any new scientific discovery, and they are going to shape the future of the next generation of microbiology. So let's start the student symposium on artificial intelligence in healthcare with this thought. I would like to invite the moderator, Dr. Sumi Dandrani ma'am, professor and the chief consultant microbiology, PGICH Noida. Ma'am, please welcome.
Thank you, Dr. Sanjana. I am privileged to have been invited here for this esteemed conference. Thank you, Rama ma'am, and the organizing committee of RAJMICROCON. So before further delay, I think we'll start the symposium. I'll invite the chairs, please. Invite the chairs on the dais. Dr. Bharti ma'am, Dr. Sonal ma'am, Dr. Maheshwari, can we have their PPTs, please? Can someone help me with the technical part, please? हाँ वही है तो सही पर वहाँ पे डिस्प्ले नहीं होगा वो वहाँ से होगा मैम ये तो सिर्फ आपके देखने के लिए रीड करने के लिए अच्छा तो वहाँ पे स्टार्ट करवा दो वो देखिए आ रही तो रमा मैम को इंट्रोड्यूस कर दो हाँ पहले उनको तो कंप्लीट करना क्योंकि उनका किसी ज़्यादा हाँ मैं So I'll first like to introduce Dr. Rama ma'am. Dr. Rama ma'am, ma'am, you can please join us on the dais. Okay. So ma'am is Dean Research and Senior Director and Professor Microbiology at National Institute of Medical Science and Research, NIMS University, Rajasthan. Ma'am has been ex-Dean Research and former Professor and Head Department of Microbiology at All India Institute of Medical Sciences, New Delhi. And she's also International Ambassador of American Society for Microbiology to India. Can we have the presentation slides for the CVs of chairpersons, please?
in the interest of time, I will introduce the chairpersons. So we have with us Dr. Sonal Saxena. She is Secretary of IAMM. She is also heading the Department of MAMC, Maulana Azad Medical College. And she has been very active in the field of antimicrobial resistance. She is WHO Fellow from, on antimicrobial resistance and conducted a lot of workshops, seminars, symposiums. A very, very active and enthusiastic person. We have with us Dr. Bharti Malhotra. I don't think anyone needs introduction for Dr. Bharti. Rajasthan, when you say microbiologist of Rajasthan, I think Dr. Bharti's name is always there. And you can relate with her name, Rajasthan Chapter also now. And Dr. Bharti has been contributing in many fields, whether it is virology, tuberculosis, and almost everywhere she is active. And I must say in COVID, a lot of work has been done. I think BSL-3 was also established by her. We have Dr. Maheshwari also with us. He is also heading the Department of Microbiology at NIMS. And Dr. Saab has been in SMS as a previous HOD of SMS Jaipur. And he has also been actively engaged in many scientific work, as well as has been encouraging younger students to really do good work and come up with scientific, interest. And so I welcome all the three chairpersons here, and we will start the symposium, which is the student symposium on artificial intelligence.
Thank you so much, ma'am. So I was very excited when ma'am told me to moderate this session. So just to begin, I feel human beings are the most evolved and intelligent forms living on Earth currently. And human brain has recently created machines which can think intelligently and which is called artificial intelligence. I feel we have been all seeing increasing use of AI in all fields around us, right from use of tools like ChatGPT for creating a travel itineraries. I myself created my travel itinerary using ChatGPT recently. To my father-in-law, who is a professor in nuclear physics, taking help of AI to create his lecture contents. And my daughter, who's doing computer science in US, using AI for writing her college projects and assignments. So we have all seen healthcare shaping in front of us, in front of our eyes, with advances in digital healthcare technologies such as AI, 3D printing, robotics, nanotechnology. Healthcare AI has become quite a hot topic recently. While we are all excited about the possible uses of AI in healthcare, there is always an indolent fear that all humans would be soon replaced by machines. Let us explore all these aspects about artificial intelligence in healthcare. And today with this student symposium, we have a very bright group of young students ma'am has gathered together, who are going to talk about all these aspects about role of artificial intelligence in healthcare. We are mainly focusing on diagnostic part in this conference. Thank you so much. With these words, I would like to invite our first student, Dr. Ritu. Dr. Ritu Joshi is junior resident second year from Government Medical College, Kota. She is going to introduce us to the topic of AI role in healthcare diagnostics. Dr. Ritu, please.
A very good morning, respected chairpersons, faculties, residents, and all microbiology fans present here. Myself, Dr. Ritu Joshi, PG resident from Government Medical College, Kota. Today, I am here to introduce you to the most special guest in the field of microbiology, or I should say healthcare, and that is none other than artificial intelligence. Yes, you heard it right. Artificial intelligence. It's going to turn stones if we used it in a right way, in a wise manner. So as we all know, that emergence of artificial intelligence in healthcare has been groundbreaking. It is reshaping the way we diagnose, treat, and monitor patients. It has drastically improved healthcare research and outcomes by producing more accurate diagnoses and enabling more personalized treatments. If we use artificial intelligence in our healthcare settings in a balanced way, our systems can become smarter, faster, and more efficient in providing care to millions of people worldwide. Here is an overview of subtopics of today's symposium, which I am going to give a little bit of idea about each one of them, which will be further elaborated by my co-presenters. So, here we start with computer vision artificial intelligence and its applications in image interpretation. The idea machines can learn and that computers can think is intriguing to many of us. When a software is used to recognize and characterize images, this application of artificial intelligence is referred as computer vision. It enables computers and systems to derive meaningful information from digital images, videos, and other visual inputs and take actions based on that information. Among the most promising clinical applications of artificial intelligence is in radiological diagnosis of infectious diseases, in which it has shown impressive accuracy and sensitivity in the identification of images, abnormalities, and promises to enhance tissue-based detection and characterization. We can also use artificial intelligence to predict pathogenesis of many diseases. And for that, we first need to understand what does horizontal gene transfer means. Horizontal gene transfer refers to the sharing of genetic materials between distant species that are not in parent-offspring relationship. It has an important role in the spread of antibiotic resistance in bacteria. So how can we understand that horizontal gene transfer events are going on? For this, we have our hero, artificial intelligence. Artificial intelligence is used to recognize horizontal gene transfer insertion sites which carries specific sequence patterns. Artificial intelligence has come up as an emerging tool for analyzing microbial metagenomic data. Metagenomics is the study of genetic material recovered directly from environmental samples such as soil, water, or feces. Since the data generated from metagenomic studies is highly complex and multidimensional, so artificial intelligence is used to ease its complexity that makes the disease diagnosis easy, accurate, and timely. So what are the ethical considerations while using artificial intelligence in medical field? They are data bias, privacy issues, accountability and transparency, and reliability and trust issues. It is crucial to note that artificial intelligence is a tool for healthcare professionals and not a replacement for their expertise. Therefore, regulatory standards and ethical considerations are vital in implementing artificial intelligence in healthcare to ensure patient safety and privacy. These are my references Thank you. Now I welcome my co-presenter, Dr. Subranil Ghosh.
Respected moderators, faculty members, and all delegates, a very good morning to one and all. Today we being standing on 2023, few years ahead of this, I suppose that our all laboratory diagnostic technologies are going to be artificial intelligence driven. And that's what I am going to talk about today. I, Dr. Subranil Ghosh, resident from Jalan Medical College, Ajmer, will be speaking on the topic, computer vision artificial intelligence. It's going to be a new frontier in laboratory diagnosis. Artificial intelligence is the ubiquitous earworm of the current era. Among the existing analytical tools, artificial intelligence has been identified as the most powerful and promising for the mankind. As my previous speaker has rightly mentioned about the computer vision, that artificial intelligence in the area of image interpretation, in which the software will recognize and then it will characterize objects, is commonly described as the computer vision. Computer vision often uses machine learning to develop software algorithms. Now, machine learning consists of three types. I'll be discussing about this in my subsequent slides. Now, computer-aided diagnosis is a computer-based application that assists medical expert in decision making. Coming to its application is the detection of Mycobacterium tuberculosis from both slide images as well as radiographic images. Other examples include pneumonia detection from CT scan and etc. Machine learning, which is a subset of artificial intelligence, consists of neural network, namely convolutional neural network, and AlexNet is the first convolutional neural network developed, which consists of overall 8 layers. Now, this picture depicts that how we are training the model, and the model is interpreting the preloaded information and they are giving us the report. Now, here it depicts that the microscope will take up the slide image and will feed the information to the system that if the slide shows acid first bacilli, then it will classify it as a tuberculosis. But if there is no acid first bacilli, it is going to classify it as a healthy sample. On the basis of this, we have seen that the training accuracy is approximately 97%, testing accuracy is approximately 91%, and when it comes to sensitivity and specificity, sensitivity is approximately 98% and specificity is approximately 83%. As I said that there are three types of machine learning. These are supervised learning, unsupervised learning, and reinforcement. As the name suggests, supervised, that means something is supervised beforehand. Here also, in supervised learning, we are pre-feeding the model with all the informations on the basis of color, shape, that how you are going to classify the organism. After that, a new data is being projected to the model, and we have seen that here we can see that the gram-negative cocci, which is supposed to be the gram-negative cocci from human detection, the machine has misinterpreted it as a gram-positive cocci, because this has not been fed to the system beforehand, that on the basis of the color and the shape. In unsupervised learning, we don't give any information to the model beforehand. On subjecting to the data, the machine itself classifies, makes a cluster of the organism on the basis of the shape and the color. And in reinforcement learning, what happens, we have two options. Either we are going to discard the sample or we are going to analyze the clinical sample. If we are going to analyze the clinical sample, then there is a resolution. If we will analyze, then it will come up with the analysis of the, resolution of the infection. And if we are going to discard the clinical sample, then we are going for the persistence of infection. That again, this cycle goes on, and that is known as reinforcement. Now, this slide shows that how the artificial intelligence computer vision flow through an implementation. Starting with the evolution of performance, as I have already discussed, on the basis of the pre-fed information, the model will come up with the result that how much it is valid or not. Following that, we will have a development of a platform for diagnostics, that is comparison of performance with current system and usability. And the very important point here is that validation, that whatever the report we are getting from through artificial intelligence and whatever the clinical outcome on the basis of that, whether it is on the positive aspect or on the negative aspect. Following that, we will go for implementation into routine clinical practice if the test or the report is at all valid, and then we need to think about the sustainability regarding the economic structure. Analysis from captured images of colonies. If you go for example, like CellaVision is a software which uses to classify morphology of white blood cells, also its ability to detect malaria parasites. Platforms are also available to classify anti-nuclear antibody patterns. It can also be used in susceptibility testing, detection of vancomycin-resistant enterococcus or methicillin-resistant staphylococcus aureus growing in screening agars, and recognize rare events in microscopic images, example, acid-fast bacilli or malaria parasite. Now, analysis of the stained microbes, as I already mentioned about the validation, that is very important. Upon validation of computer vision, there is a detection of protozoa in trichrome stained fecal smears. Specifically, computer vision is ideal for screening large number of images, but that too for rare but defined events, which is how applied computer vision to ova and parasite detection. This approach uses the strength of both computer and humans. That means it's going to have a collective contribution both conventionally and technologically. Now, this is how the artificial intelligence will project us with the report with a probability that it shows that the first person 97% probability of gram-positive cocci in change, suggesting it's a streptococci. It will be exactly how we'll be seeing under the microscope. Now, as artificial intelligence being on today's era is a very debatable point, I am going to talk about the positive aspects of artificial intelligence, as because these are going to change our life in very wide perspective. Now, AI is increasingly becoming an important component of clinical microbiology informatics. Across all the fields, researchers, microbiologists, laboratorians and diagnosticians are interested in AI-based testing because these solutions have the potential to improve the test turnaround, and that is the most important thing. As we will get the report faster, we can treat faster, clinician can treat faster. Also, the quality and the cost should be maintained. AI diagnostics include genomic information from isolated bacteria, metagenomic microbial findings from primary specimens, mass spectra captured from cultured bacterial isolates, and large digital images. Now, if we compare the computer vision on 2023, we can very much correlate this with the polymerase chain reaction in the year 2020. At that time also, we are thinking that whether how much it will help us in diagnosis, and today PCR is used like anything, and widely using is very much acceptable. So I think that computer vision 5-10 years down the lane will be very much acceptable to the community. So clinical microbiologists that train 20 years from now will recognize that how important it is. The take-home message that I want to say is that as computing power will come up, more and more ideas will come up, digital imaging, slide scanning, bacteriology automation and digital storage become more and more accessible. So, all the companies, creative laboratories and individuals will come up with innovative ideas so that we can rectify the errors within that, and we can fine-tune that all this. Artificial intelligence in general and computer vision in specific are emerging tools that clinical microbiologists need to study, develop and implement in order to improve clinical microbiology. And that's how that computer vision represents the next frontier in laboratory diagnosis. These are my references. Thank you. Now I invite my next speaker, Dr. Riya Mukherjee.
Good morning, respected teachers and my fellow colleagues. Today, I am Riya Mukherjee, resident of SMS Medical College, Jaipur, is going to present a symposium before you all. And my topic is Artificial Intelligence for Aid in Radiological Diagnosis of Infectious Disease. As my fellow colleague, Dr. Ritu Joshi, has mentioned earlier that it is indeed the most promising in the field of radiology. So, starting with some basics, radiology is a branch of medical science that employs radiation to produce medical imaging such as X-ray, CT, ultrasound and MRI images. So why AI for aid in radiology? Because it can automatically find abnormal patterns in picture data that can help clinicians to diagnose patients. So what are the benefits of using AI in radiology? It gives more accurate classification, it helps in generating 3D models, enhanced analysis, and it gives quick results. So now I will be talking about AI application in infectious diseases. So AI-powered thermal imaging tool for temperature screening. During and after COVID era, we all have seen in places like airport, shopping malls, or colleges, schools, officials, they are using thermal imaging tool. Yes, it is based on AI only. So it is based on individuals are remotely monitored as they pass the screening checkpoints. The operator looks at the color monitor that shows the audible and visible color alarms and when the individual shows out of norm temperature. And then the operator will identify the individual who has elevated body temperature and send that person to a separate line. Now some machine learning techniques. First I will be talking about high resolution melting curve analysis. It is based on the principle of detection of allele expression imbalance. One allele of exonic SNP, that is single nucleotide polymorphism, chosen as a marker is completely complementary to one allele of the single label fluorescent probe. And the result is the heterozygous individuals are distinguished by the differences in their melting temperatures. Research was done in the year of 2011. It was detecting differential allele expression using high resolution melting curve analysis application to the breast cancer on SHEC2 gene. And the result was the high sensitivity of HRM for the detection and quantification of differential allele expression. HRM was also able to detect DAE associated with nonsense-mediated mRNA decay in LCL, that is lymphoblastoid cell lines carrying nonsense mutation in cell cycle checkpoint kinase 2 gene. Now I will be taking support vector machine algorithm. What are the uses of SVM? They are used for linear or nonlinear classification, text classification, image classification, gene expression analysis, and anomaly detection. The main objective of this SVM is to find the hyperplane in an n-dimensional space that can separate the data points in different classes in the feature space. If the number of input is 2, then the hyperplane is just a line, and if the number of input is 3 or more than 3, then the hyperplane becomes 2D plane. And the combination of support vector machine and HRM could accurately identify isolated bacteria. Now, artificial intelligence in pulmonary tuberculosis. In the picture, we can all see it is the application of artificial intelligence in digital chest radiography, reading for pulmonary TB. AI uses predictive algorithms for diagnosis of pulmonary TB. Those are decision trees, SVM, that is support vector machine, naive Bayes classifier, genetic algorithm, and fuzzy algorithm. Artificial neural network. A study was done in the year of 2019 for the prediction of TB disease. Why ANN? ANN because it plays an effective role in early diagnosis of TB that might be applied as a supportive tool. And what they found is that the Mycobacterium tuberculosis positive or negative with an overall accuracy more than 94%, and accuracy of the test and validation was more than 93%. Now I will be talking about artificial intelligence in COVID-19. In the year of 2021, a research study was done using artificial intelligence to assist radiologists in distinguishing COVID-19 from other pulmonary infection. So it was based on segmentation model. Based on ResUNet with original CT images, and they trained ResNet 50 classifier with the lesion mask, pneumonia, non-COVID pneumonia, and COVID pneumonia. And the final probability was given in terms of confidence, which includes class prediction, proportion, double lung, and lung periphery. Now, deep learning based automatic detection, that is DLAD. A study was conducted on this algorithm for chest X-ray abnormality, and the objective was to evaluate of DLAD performance compared to that of radiologists in the standard clinical practice. A total of 127 images were taken, and in 127 images, 40 with the finding and 87 without any finding. So DLAD correctly identified 37 images as abnormal and 56 images as normal. So lastly, I just want to say AI-based approaches, including deep learning, radiomics, and other conventional machine learning methods applied to TB, per se any other disease, provides a self-driven, convenient, and time-saving strategy to improve diagnostic efficiency and accuracy and outperforming radiologists. Thank you very much. And these are my references. Now I would like to call Dr. Ayushi.
A very good morning to everyone. I am here Dr. Ayushi, resident Dr. SMS Medical College, Jaipur. And I'm going to talk about artificial intelligence to predict pathogenesis and horizontal gene transfer events. So we know that evolutions and innovations in having genetic basis are transmitted vertically from parents to offspring. However, genetic information can also be transmitted when there is no inheritance. So here comes the picture of horizontal gene transfer. So what is horizontal gene transfer? It is sharing of the genetic information between organisms that are not in a parent-offspring relationship. This is a widely recognized mechanism for adaptation in bacteria. It is prevalent in prokaryotes and leads to spread of antibiotic resistance and emergence of pathogenicity. So in the picture we can see there is a difference between vertical transmission and horizontal transmission. In vertical transmission, whole genome is transferred and there is inheritance of the parental traits, whereas for horizontal transmission, the transmission leads to transfer of only few genes and there is introduction of new traits to the organism. So these are three mechanisms, three most recognized mechanisms for HGT: horizontal gene transfer, transformation, transduction, and conjugation. Transformation is nothing but transfer of the cell-free or naked DNA from one cell to the another. Transduction is the transfer of genes from one cell to another with the help of bacteriophage, and in conjugation, there is a transfer of genes between cells that are in physical contact with one another. Now, what are the consequences of horizontal gene transfer? This is a dynamic and persistent phenomenon that can have immediate to delayed effects. This is involved in bacterial genome evolution, genome damage prevention, antibiotic resistance, virulence, and compatibility to environmental stresses. HGT allows different species to share genomic fragments, thus creating a complex network among different species and evolution of bacteria. This is crucial in development, emergence, and recurrence of various human-related diseases such as cancer, genetic, metabolic, and neurodegenerative disorders, and can negatively affect the therapeutic outcome by promoting resistant forms. Thus, the genomic and biological impact of HGT could be more widespread in the tree of life than initially thought. So we know that WHO has recognized antimicrobial resistance as one of the top 10 global threats to mankind, and this is going to be the most emerging and burning topic in near future. So to understand antibiotic resistance very well, we should know what HGT is. Resistance are of two types, intrinsic and extrinsic. The intrinsic one is mostly inherited, whereas the extrinsic one are mostly generally obtained by horizontal gene transfer, and this includes plasmid encoded and transposomes mediated antibiotic resistance. Now, the detection of HGT and mechanisms responsible are obviously of very prime importance, as we know that this is responsible for the genome evolution and the adaptation which bacteria does. So there are two classical ways to identify candidate HGT using AI. So firstly, there is to study the intrinsic one, so that is the GC content, codon usage distribution, and the extrinsic one will be using percentage identity, blast E values against other species. So these are the two main ways to identify candidate HGT using AI. So what is mobile genetic elements? So these are mostly transposomes. These are the main agents of HGT. These transposomes, as shown in the picture, they have a special sequence feature at the end, and this makes transposomes easily transferable across cells by plasmids and phages, and therefore it is possible to recognize the HGT insertion sites according to the sequence features at the sites. So we can see in the picture also that transposomes have the special sequence feature at the ends, and this makes it very easily transferable across different cells, and therefore it can be used to recognize HGT insertion sites.
Now what is deep learning? To understand deep learning, we should know what AI is. AI is a technique which enables machines to mimic human behavior. Machine learning is a subset of AI which uses statistical methods to enable machines to improve with experience. And deep learning is a subset of machine learning which is going to which makes the computation of machine multi-layers neural network feasible. So deep learning is a very powerful method to extract features from the DNA sequences. It is a type of machine learning algorithm which is based on artificial neural networks. And one such tool is DeepHGT, which is the first deep learning model that is used to recognize HGT sites directly on the bacterial genomes. As we know, bacterial genomes, to understand these bacterial genomes, it is a very tough task. So deep learning is going to help us as an AI tool to easily understand and extract DNA features from the DNA sequences. Now, one such tool is Dark Horse. Dark Horse was initially developed to identify HGT in bacteria, and this is a software. So this proposes a lineage probability index score which is based on the taxonomic ranks of the candidate HGT from BLAST against the protein databases. So we know that the gold standard to predict HGT is phylogenetic methods, but this is very time taking. So the AI or HGT index based methods are an interesting method so that we can quickly identify the candidate HGT, and which is going to help us in near future. Alieness is one such user-friendly web application. It requires the BLAST results against any sequence library, and this is also applied on any genome of interest. So this is a very variable and versatile web application which is applied to any donor and any recipient taxonomic group, and is very helpful to identify the candidate HGT across the tree of life, and will contribute to assess more globally the evolutionary significance of HGT. Now this picture shows how the gene transfer occurs via HGT. There are the three mechanisms as I've told earlier: transduction, transformation, and conjugation. After the HGT, the new genome forms, and these genomes have different islands, different genome islands like pathogenicity, symbiosis, metabolic resistance, and fitness islands. So this is going to make the bacteria evolve more easily, and this will help the bacteria to adapt to the different environmental conditions, and therefore it can survive the environmental stresses which the bacteria faces. Now this picture shows how time consuming was the genome sequencing type. So this was a previous technique, and now also it is used. So this is a time taking one, whereas the computational detection can be a very time efficient method to be used, and this is a precise methodology employed to evaluate the machine learning algorithms. So this will finally lead us to replication box, codon, coding regions, conserved sequences, so various such kind of genes and repeats. So I would like to conclude my presentation by saying this: the selfish environment meets the selfish gene. Yes, it was rightly said by Monaco that the selfish environment meets the selfish gene, as we know that the distant species are connected by a horizontal gene transfer. The resistance pattern can be spread, and there is emergence of pathogenicity and resistance across various species, from prokaryotes to eukaryotes, and this is going to be a most important method for resistance in near future. Thank you so much. And I'm going to request Dr. Shikhir to continue with Artificial Intelligence Symposium.
Thank you, Dr. Ayushi. A very good morning to all the respected faculties and delegates present over here. I, Dr. Shikhir Malhotra, Junior Resident from Department of Microbiology, AIMS Jodhpur, is going to shed a brief introduction and just a food of thought about the AI in conjugation with a new field coming in metagenomics, that too directly from the primary specimens. So what is metagenomics? As we all know, it is the study of and structure of function of entire nucleotide. This entire nucleotide sequence has in turn produce the data in kilobytes. So just looking up at this image, if we see, we have obtained the specimens from the human in any form, whether it be the blood, the pus, or any sample, and in turn we have extracted the DNA, and we have in turn come out with the nucleotide sequences. Now in this nucleotide sequences, we already know the data is produced in almost kilobytes to tetrabytes, and human mind sitting over here would create a lot of ruckus. So here comes our AI friend presenting us with the library preparation, Illumina sequencing, the nanopore sequencing, the ultimate structures which required supercomputational powers. So these duo, that is the microbiologist and the artificial intelligence, presents as the detectives. These two of them have their own role in conjugatingly giving this result. So just looking up at this, from the specimen we have received, as discussed earlier, the AI or our friend is here for data categorization, clinical data evaluation, and categorizing them as to what your needs are required. And what about we, the microbiologist over here? We are here for doing the in-hands work, that is the extraction and amplification, and looking up at this data entry. Once this data is processed, as what we had discussed earlier, the data has been generated in tons of work. And is this really going to be useful? So the preparation from library, pathogenic identification is done by our friend artificial intelligence, whereas the role of microbiologist in this is that the isolates which we have recovered, do they really have pathogenic significance to the human? And if yes, what was their phylogenicity or their mutations with respect to that? So as earlier discussed by my fellow colleagues, there are different domains of the artificial intelligence dealing with reasoning, representation, learning, processing, and perception. And in conjugation with these, let us just have a look at how this AI can help in the terms of metagenomics. So as discussed earlier, ANN in simple word is nothing but the neuron for the artificial intelligence. The data you feed to it, the data you will get back through it. Logistic regression, very most important things. One, we have categorized the metagenomic data, whether this data has a single dependence or what we call the univariate analysis or the multivariate analysis, that is also equally processed by the artificial intelligence. Thirdly, it hierarchically clusters and gives it the phylogenicity relation. So artificial intelligence, nothing but the modus operandi for detecting pathogens. We all know that the metagenomics have evolved from the simple word, or what we are now looking is the whole genome sequencing. The data as told by G et al. In 2017 say the data obtained from this NGS or WGS is highly complex, high through output, and a large data volume which need to be computerized. So what is this? How they can help us in detecting? One is tracing outbreaks by the artificial intelligence. Study by Sudhakar et al. In 2021 had used this conjugation with detection of Salmonella typhimurium, and they have wisely associated and found out where the clusters were originating from. Clinical utilities, will be just looking up at the further case reports and detecting the food, agro, or waterborne pathogens. So this was a review done by Patel et al. Regarding the monkeypox infection. The metagenomics has provided us with a lot of data. Now, with this respect, what is the importance to the human is reviewed in this, that is monitoring and predicting, tracing and tracking the cases of the monkeypox, case surveillance, prediction of the protein structure of the variants. As we know, there was East African clade and the West African clade. So what was the clade differences, which are also pointed out with the metagenomics? Then reducing the burden from the medical practitioners, as what we have discussed earlier, the diagnosis, the vaccine development, and the most important thing in our world, to control the spread of false information. Another study by Farron et al. By JCM had used the automated scoring for chromogenic media using the WASP lab image software. So almost 57,690 swabs were identified over here, and MRSA was detected by the artificial intelligence, and it was found out that almost a sensitivity of 100% with a specificity of 90.7% was in concordance with that of the results. So here we can look that the picture 1 shows the negative without any growth. The picture 2 is showing the MRSA colonies on the chromogenic media, and picture 3, if we zoom up, shows a white or pale colonies. So the true detection by AI had played an important role. Similarly, in another study had used for VRE in their detection, and almost the concordance result of 104,000 samples were used, which had found only discordance in 10,000 samples, giving automation a sensitivity of almost 99% and a specificity of 89.5%, as again visible in this chromogenic media. And in last, there was a review by the antibiotics. It's not only the data which has automatically been generated by the artificial intelligence. The further AMR outcomes which are coming, that is the continuous surveillance, new drug development, identification of these coming variants are equally predicted by the artificial intelligence. So in just to conclude, artificial intelligence and metagenomics, this is our way forward. The metagenomics or whole genome has opened up a whole new gateway for us. And this gateway to analyze what is useful is being done by the artificial intelligence. Thank you. And with this, I'd like to call Dr. Amit to shed some light on the ethical standards.
Good morning, everyone. Myself, Dr. Amit Gupta, PG first year resident of Department of Microbiology from Sardar Patel Medical College, Bikaner. Want to discuss our last topic of symposium, that is ethical use of artificial intelligence in healthcare to augment, not to replace human efforts. What are the ethical use? The central values upon which all the ethical principles rest are what? These are inherent worth of humans and human dignity. What are the various problems and challenges of artificial intelligence in healthcare? These are broadly classified into two categories. These are ethical and legal. But here we have to emphasize mainly on ethical problems. These are regulation, privacy, transparency, and bias, and many more. What are the key ethical principles? These are based on the 4 principles of oath which we take. Which we took in our previous days of MBBS, first year MBBS, that is known as Hippocratic Oath. These are non-maleficence, beneficence, justice, and respect for patient autonomy. What are the basic ethical requirements? First is protect autonomy. First, patients are fully informed about how artificial intelligence will be used in their healthcare and to obtain their informed consent. Second is ensure transparency, explainability, and intelligibility. Communicates how patient data is collected, stored, and utilized by artificial system, ensuring patients understand how their information is being used for healthcare purposes. Promote human well-being, human safety, and public interest. The use of artificial intelligence in healthcare is to bring a positive change in the society by improving patient outcome and enhance safety of the patient. Thus, it is to ensure that it must be serving for public interest. Foster responsibility and accountability. Who bears the responsibility if anything goes wrong? It defines that it clears roles and responsibility of all stakeholders, including developers, healthcare professionals, administrators, and regulators involved in the development, deployment, and operation of artificial intelligence system in healthcare. Promote artificial intelligence that is responsive and sustainable. We have to design artificial system in healthcare in such a way that it has to bring a positive impact on evolving medical knowledge, patient needs, and changing healthcare environment. Thus, it is to ensure that it must be relevant and effective over time. Ensure inclusiveness and equity. Data which is used for artificial intelligence in healthcare must be diverse and representative of different population, including underrepresented groups, to avoid bias and disparity. For example, if we have to diagnose cancerous lesion in black-skinned people, but the data fed in software or library of artificial intelligence is of white people, it might lead to disparity in the results and increasing the risk of their lives. What are the ethical challenges? First, assess whether artificial intelligence should be used to improve patient outcome, enhance diagnosis accuracy, reduce cost, and increase efficiency. Artificial intelligence is the digital divide. This artificial intelligence in the modern era basically depends on the internet access and smartphones. But the developing countries like India, people or patient does not have a smartphone or internet connectivity, then their data must not be incorporated into artificial intelligence system healthcare for health. Therefore, it leads to disparity in the result. And patients are also frightened to give their consent for their data to transfer to anyone. That is data privacy, another concern for artificial intelligence. Data collection and use. Data collection and use is as a broad term for medically broad term, which include data from the genomics, data from non-health data, medical record data. Therefore, all these collection is known as biomedical health records. Therefore, we have to must ensure that while collecting data, it should be safe, transparent, and data quality is good. Fourth, who bears the responsibility and accountability for decision making? It depends on all the algorithm developers, professional institutes, and responsibility to make decisions based on ethical principles and to prioritize patient well-being. Autonomous decision making. Autonomous decision making is enhanced healthcare decision making while uploading ethical standards and respecting individual rights and preference. Bias and discrimination. Biases depend on the gender, race, and sexual orientation. For example, racial bias in USA and Africa leads to challenges to artificial intelligence in healthcare. Risk of artificial intelligence technology. Cyber theft are the biggest challenges for artificial intelligence in the modern era. For example, cyber attack on UVM Health Network and Nebraska Medicine Obama health attack lead to a shutdown of healthcare for few days, due to which there is a loss of money because of there are a lot of rescheduling and appointment rescheduling. Impacts of artificial intelligence on labor and employment. Definitely artificial intelligence decreases the labor, staff and jobs because of the automation of routine tasks, enhanced diagnostic and telemedicine. What are the challenges in commercialization for artificial intelligence for healthcare? These are data privacy and security, reliability and safety, data transparency, accountability and reliability, cost. What is the most significant negative aspect of artificial intelligence in healthcare? The feelings of compassion, empathy, emotions, love makes doctors and nurses superior than robotic machines, which do not have these feelings, because these feelings are very necessary for the healing process of the patient. Patients such as children, psychiatric patient and obs and gynae patient will lose empathy and kindness and appropriate behavior when dealing with robotic physician and nurses. Children and psychiatric patients may have anxiety and fear due to the robots, and it makes an uncomfortable environment for the patient. Robots used in COVID-19. In India, robot name is Mitra, and Italy robot name is Tommy. These robots help patient in COVID-19. Artificial intelligence to augment, not to replace human effort. Artificial intelligence must be used only for screening and not for confirmation. Confirmation is only done by humans, because humans have the capacity to think and make a decision in critical condition, which is not possible by the machines. In the last, I want to conclude that by a famous quote known as Fundamental Theorem of Informatics given by Friedman in 2009. Don't make use of excellent technology, make excellent use of technology, which means computer can never be superior than human brain, because computers are invented by human brain and not vice versa. But when computer skills are added with the human brain, then it is always superior than human brain alone. These are my references. Thank you.
Thank you all of you.
Yeah, ma'am, you continue.
So I like to compliment all the students. I'll request all the students to please come on the stage, come on the podium. And I want to make all of you aware that they have been working hard. We've been working with them, trying to moderate the sessions, and for more than a month, under guidance of Rama ma'am and Dr. Sankalp, who's also in NIMS. Sir is data analyst here. And we've had a wonderful session covering most of the aspects related to diagnostics in use of artificial intelligence in diagnostics. With these comments, I would like the chairs to give their comments, or even ask if they want to ask some questions from either of the students. Dr. Sonal ma'am.
Thank you, Sumi. I think it was very comprehensively covered, and we all will agree on it. This is something which we are all still learning. And when computers came, I still remember when I was a senior resident and the faculty would tell us prepare a lecture. It used to be a daunting task for both the faculty. You remember Sumi was PG in those days. So similarly, I think then we learned Excel, and Excel still we are learning, I feel, some or the other new feature. So this is a very, something which is coming on horizon, and we are all going to learn a lot. One of the very major aspect of AI, which I feel is going to be a game changer, is its predictive ability. Just like in COVID, we saw people were able to predict that yes, by the end, this variant has come. So by the end of this month, we will have these many patients, these many samples. So be ready with this much of inventory for testing, this much of treatment strategies, and this should be your preparation for treating them. Similarly, I think a patient comes into our ICU and we come to know that if he's got these parameters, these are the chances that he's going to be sick with sepsis or die with sepsis. I think that is going to be a game changer. The other thing with the lot of work going on is in spearheading development of new antibiotics. Because till now the pipeline for antibiotic is dry and it takes a long time for one antibiotic to reach phase 3 trials. So a lot of machine learning and AI tools are being used now to develop newer antibiotics to see the efficacy of antibacterial peptides and bacteriophages, and that is, I think, going to be another game changer with AI. Thank you.
Okay. I would like to congratulate Sumi and Dr. Sankalp for really coordinating, moderating this excellent session. The students have really presented very well, and it was a very comprehensive presentation and covering all aspects, whether it be metagenomics or be the smear diagnostics or the radio diagnostics, and when to use and when not to use, and the ethical issues. Of course, now it will be very fast. The moment you give an assignment for the students, it will be very fast to be ready with the presentation. They are going to use this left and right as the chat GPT, and very soon the issue would be the plagiarism. Everybody would be coming out with the similar script. So it will be a smart tool to handle, but you will really have to handle it smartly so that you are not falling into the trap of plagiarism. So I really congratulate you, all of you.
Thank you, ma'am, for your comments. Coming to plagiarism, I was just, while preparing them for this symposium, we came across a lot of issues. Artificial intelligence, in fact, is so intelligent that it is able to create false citations. It is very difficult to trace the citations to be false. There have been major issues. In fact, my daughter had a concern. Most of the Indian students have a problem with English in US. So she took help of ChatGPT to prepare one of her assignments in English. And the teacher came to know that she's taken help. And she was really scared because this is becoming a very big issue in medical teaching as well. We should all be aware that students are more likely to take help of these tools which are so easily available and accessible. And we should all try and teach them and train them that in spite of all this availability of all this data and all these tools, we still have to be genuine to what we are writing and what we are researching on or what we are studying. So I feel it is a big issue. With the power it comes, the responsibilities also have to be taken into account. Sir, Dr. Maheshwari, sir.
I congratulate to all the students and Dr. Sumi for very laborious and 1 month hard working on this new topic. In my opinion, what is the basis of artificial intelligence? What is this basically? So, in my opinion, actually it is a branch of computer science which aims to create the machines and develop the software which can perform the various tasks which otherwise normally that task required the human intelligence. So this human intelligence gets replaced by the artificial intelligence, such as learning or reasoning or problem solving, etc. So this diagnostic part in the healthcare, it is nicely covered by these students. Apart from that, as this madam told that it is also helps to develop the new antibiotic molecules. Apart from it, not only to develop the new molecules, it also helps in to test their efficacy and safety in silico. That means the efficacy and safety, it is to be tested in the computer simulation before going for the in vivo or into the living beings. So that is also the role of the artificial intelligence. Apart from that, it also, as we discuss in between, also it helps in the prediction of the diseases. as you learned few days earlier that WHO or some news they are coming that one another outbreak is likely to become. So here also there is a role of the artificial intelligence by which we understand the various microbial interaction, applying this knowledge of the ML and analyzing the epidemiological data and various reports, environmental data, surveillance data. So all these they are intermingled and they are all these data they are treated by the artificial intelligence and we can detect that this outbreak is likely to be occur. Apart from that, it also helps in the interaction between this host and microorganisms. It's shaping in the field of the microbiology. It is a artificial intelligence it provides by providing the faster, most accurate and comprehensive solution for the various problems related to the microbiology. And it increase the skill and knowledge of the microbiologist for better decision and improving this healthcare, human health, well-being. So apart from that, we already covered this ethical issue, but some other challenges are also related with this AI, such as apart from this ethical issue, this data quality issue, availability and interpretability and explainability. These are the some issues, and human-machine collaboration it is also an issue for related to the AI. So that's why it is important for the microbiologist to well understand the principle and application of the AI and to collaborate with this computer scientist and other expertise to ensure that the responsible and beneficial use of this artificial intelligence in the field of microbiology. So again, I congratulate to all.
Thank you so much, sir. With these comments, I would also like to highlight, I was going, while preparing them, I listened to a lot of seminars and webinars. There was one webinar in which how artificial intelligence has to be very carefully used, how it can give wrong results. So there was an example that they use artificial intelligence for creating driving softwares in which a car could automatically drive. But there was a major flaw in that, because the stop sign which was fed into the software for artificial intelligence, they were not able to detect the graffiti done on the stop sign. So the artificial intelligence software was not able to actually detect a stop sign on which some graffiti had been done, which led to major accidents and it had to be recalled. So a machine will only interpret what it has been fed. We should all be very careful on that. Since this was brainchild of Dr. Rama, ma'am, one or two words from you. Thank you. Okay. Thank you. Thank you so much. I'd like to thank the chairs. I'd like to thank all the students and Dr. Sankalp and Rama ma'am also.
So thank you all dear residents and the moderators. So now I request all the chairpersons to please present a memento to our moderator for this session. So I request Bharti ma'am, Sonal ma'am and Maheshwari sir to please present a memento to Sumi Nandavani ma'am. Thank you, ma'am. Please present a memento to Mr. Sankalp as a moderator for this session. Now, I would like to call upon Dr. Rama Chaudhary ma'am and Dr. Shama ma'am to please come on the dais and present a memento as token of respect to our chairpersons. Okay, fine. So, ma'am, I request Rama ma'am to please present a memento to Sonal Saxena ma'am. I request Shama ma'am to please present a memento to Bharti Malhotra ma'am. Requesting Rama ma'am and Shama ma'am also to present a memento to Maheshwari sir also. Yeah, yeah. Students के साथ. बस अभी हाँ. So we just have a group photograph with the students. So I request all the chairpersons and. Mr. Sankalp, Mr. Sankalp Sharma, I think he is not here. So thank you all the chairperson, residents and moderators for the first session, student symposium. I will continue. Oh, ma'am. So after this very informative and thoughtful provoking student symposium, we now go for our next session 1. So I again welcome you all. So a very warm good morning to everyone. I, Dr. Mamta Lamba, Assistant Professor, Department of Microbiology, SMS Medical College. Welcome to all our esteemed guests, teachers, seniors and delegates all over the Rajasthan. So today we all are here gathered for the second RAJMICROCON, as we all know, with the theme Microbe Invisible to Invincible. So the theme itself says we all know that the microbes are invisible. They are too small to be seen by the naked eye, but they are invincible. They are too powerful and too strong to be defeated, so we should not underestimate the power of these microorganisms which we are dealing in the microbiology. So with this theme, let me first start our scientific session on the emerging infections. For this, we have the chairperson, Dr. V.M. Katos sir and Dr. S.K. Singh sir. So, Dr. V.M. Katos sir is the President, JIPMER Pondicherry, and sir is also chairperson for the Lepra Society, editor, Indian Journal of Leprosy, and former Director General, ICMR. Sir will be chairing the session from there only, and the next chairperson, Dr. S.K. Singh sir, sir is the head of the Department of Microbiology and senior professor, Department of Microbiology, SMS Medical College. So the chairperson will be continuing from there only. So let me first invite our first speaker, Dr. Bharti Malhotra ma'am, senior professor and in charge VRDL lab, Department of Microbiology, SMS Medical College. Ma'am will brief us about the genomic surveillance of COVID-19. Welcome you, ma'am. So I request the chairperson to introduce our speaker.
चार फोल्डर है इसमें से
मैं खोलती हूँ.
हेलो हेलो.
खुला नहीं. तो फिर कैसे होगा? अभी वो शायद वो उधर से स्लाइड चलाई है इसलिए नहीं चल रहा होगा.
नहीं मैम यहीं से चल जाएगी.
Again, you know that Dr. Bharti doesn't need an introduction because already introduced. She's the president of the Rajasthan Chapter at the moment and a very accomplished microbiologist. I think was contributed to the growth of microbiology, at the development of the many things in the department. It itself was a big department which has evolved over a period of time, but her contributions to tuberculosis, virology, and several areas which she chose are well known. Dr. Bharti, go ahead. Yes.
It's hanging. Anyway, by the time he fixes up. So it's a great honor to have introduction by Dr. V.M. Katoch, who's been my mentor, who's guided me throughout my life. And is still, and lucky to have him in Jaipur now and guiding in person on a regular basis. Thanks to him, I've learned all the nuances of microbacteriology. And due to his initiation, I was into the virology field. And with his support, we established the VRDL lab. And now it's a state VRDL lab. And because of that VRDL network, we could manage the COVID pandemic very well. And this presentation is dedicated to my mentor. And I would like to thank Dr. Rama ma'am and the organizers for inviting me for the talk. There is some problem in... क्या हुआ? पहले तो चल रहा था. फिर तो प्रेजेंटेशन वहाँ से आप पहले कैसे चलाई थी? इसी की ड्राइव में थी? दूसरा लगा दो. क्या हुआ? तो वो प्रेजेंटेशन कहाँ से लगी है? वो उधर से हो रही है. ओके. तो लैपटॉप दूसरा लगाओ. नहीं तो वो स्लाइड कैसे चेंज होएगी? स्लाइड चेंजर दे दो. वहाँ से प्रोजेक्ट कर दो. स्लाइड चेंजर दे दो. और वो पॉइंटर है, पॉइंटर भी दे दो, स्लाइड चेंजर भी दे दो. Sorry for the delay, but it's not at my end. So. तो ये लैपटॉप में आएगा, उधर से देखेंगे फिर लैपटॉप. हेलो. Well, we've all faced the pandemic, and while we were having this pandemic, what came to our rescue was the guidance from ICMR, the DHR. We had a good VRDL network, and thanks to the VRDL network created by Dr. Katoch, and we were all well equipped to handle that pandemic. And very soon we had the diagnostic kits available with us, the guidance how to collect samples, transport them, and where to send the samples. And very soon, within a few days, by the 1st February, our center was recognized to be the testing center for COVID. And then later on, we did maximum up to 10,000 samples a day in the peak time of COVID testing. And then eventually, we started the sequencing. So the role of the sequencing was, to predict what. So though of course now the COVID has declined, but what we faced through this, these are the lessons what we learned and how, why and how it was important to do the sequencing, how it helped us to control the outbreak and the pandemic in the state. We were the center for the state, and then we could manage to guide the public health people to control the outbreak. So why we did the whole genome sequencing? It is important to do the whole genome sequencing because to identify the mutations and to do the molecular epidemiology of the virus, so we could track and trace the virus and study the transmission dynamics. And because of these mutations, we could identify whether these new mutants are escaping the immune response and whether the vaccines are effective or not. And these, when the Delta wave came along, we could identify it quickly and help the public health authorities. ये आपका slide कहाँ से change होगा? ये move नहीं हो रहा slide. So during this whole process, WHO came out because initially the variants, there were different way of classifying, and over the period of time, various classifications emerged to classify the various mutants. And there was the Pango lineage, which was the method of classification. Then GISAID came out with this classification. So the various classification and next strain classification came along, and we had these various variants. And WHO, now to make life simple, came out with the classification of variant of interest, variant of concern, and variant for monitoring. And these were dynamic classifications from the WHO, because, for example, when initially it was the alpha strain which came from UK and which was causing, increase in transmission. But very soon that disappeared and it was replaced by other strain. So then it was no longer a variant of concern. So this was a dynamic classification. Yeah. Change कैसे करेंगे? Well, this was not by design that I wanted to be on the stage for long, but it was no fault of mine. So we had these variant of concern, VOC, variant of interest, and variant of under monitoring, and variant of high consequences. And this was by definitions given by WHO, and this was a dynamic classification, because over the period of time, when the variants spread and immunity developed, then they were no longer in circulation and the new variants emerged over the period of time. So we will go through the journey which we faced during last 2-3 years and the havoc it caused to us. So the VOCs are named as VOCs because they have impact on diagnostics, the treatments and the vaccine, and interferes with the diagnostic test targets. Very frequently we saw that the kits which were used effective earlier were no longer able to identify the virus. So this was affecting the diagnostic target of the kit. So we had these various labels, Delta and the Omicron. All these were the variants of concern. And there were increased transmissibility, increased disease severity, and increased hospitalization and diseases. So WHO had these pango lineage. If it was, the delta was the common, the slang language, but it was the technically by the pango lineage, it was B.617.2 and the AY lineages. So here the spike protein mutations were seen, and the next strain clade, it was again a different classification. So many times it was confusing for people, but it was better to stick to one classification so you could understand what is happening in the. ये अटक गया आपका, किधर से चेंज होएगी? चलो आप चेंज कर दो. So similarly, there were variant of interest, so you had to monitor them, that these something which is emerging, and then if you keep on tracking, so sometimes then they jump the category and became variant of concern. Next. So similarly, we have had these epsilon, zeta, iota, and recently we had B.1.16, XBB 1.16, which was the Arctus. Next. So similarly, there were many variants which came along and then passed away after some time. Next. So similarly, we had these variants of under monitoring. B.1.640 was one of the variant under monitoring. Next. So variants of high concern, because it was very important to send the message of how to prevent them, and they demonstrated failure of diagnostic tests, and there was reduction in vaccine efficacy, and there was reduced susceptibility to multiple therapeutics. So these variants were known as the variants of high consequence. Next. So then XE emerged over the period of time. Next. And then the classification, as I told you earlier, there are three types of classification. Pango is the phylogenetic assignment of named global outbreak lineages, and this is one way of classifying it. Next is the next strain classification. So you will, when you are seeing that various types of classifications are coming up, so there is some variation, and they have these kinds of generally we are using the Pango classification. Next. So this is a article which is describing about the Pango lineages, how they are identify various lineages and how they are emerging. Next. So these again is showing the Pango lineages nomenclature. Next. So this is just depicting the GIS8 database. There's a GIS8 database where all the sequences are deposited, and then where if you go through that, then you can see the evolution of the virus and how over the period of time, which variants are emerging. Next. So GIS8 classification is again based on major clades and based on the marker mutations from S to SL and then G, GR, GH. Depending on over the period of time, mutations occur, and then they evolve from G to GH, GR, GV, GR, GRY, and so on and so forth. Next. So these are the various marker mutations in the GIS8 clades for the various clades. Next. So if you look at the, again as I told you, if you look at the GIS8, you can see so what was initially here, the clade over the period of time, it changed, and this is different clades prevalent in different parts of the world. Next. So this is, you can see the in May 2020, it was the alpha which was predominant, and from the June onwards, the delta started emerging, and it became the predominant strain for many months' time. This is the time of the second wave where very high mortality and very high hospitalization rates were seen. So we could identify these delta strains emerging in our state in May and June period. This is the time which we started sequencing, and we could alert the authorities, and they could really take precautions about this, and then they were on the high alert. Similarly, in December 21, we came across few strains of Omicron, and very fast initial Omicrons were from the foreign travelers, and later on we found that they were occurring in patients who were coming from, who had no history of travel, and we could, alert the clinicians and the public health authority. Initially, we isolated these patients, but once we started finding them in patients without history, so the strategy of public health changed. Next. So these are the changes over the period of time. Next. Next, this is similarly, you can see the changes happening over the time. So there are many, because of this need of the many networks emerged, which were genomic surveillance networks, like there was a United Kingdom network, there was a South African network, of course, and this during this time. Initially, the ICMR, we were sending our samples to NIV, then we started doing it at our place, then the INSACOG was formed by the Government of India, DBT, and in collaboration with NCDC, where all the sequences were supposed to be submitted in their platform, so then they could also monitor the emergence of the various variants in a more organized fashion. So the very organized fashion, a system of INSACOG was developed, where weekly we were having meetings and we were asked to upgrade our sequencing depending on the positivity, or downgrade it depending on if it's the same strain which is prevalent, to have the cost effectiveness of the whole procedure. Next. So this is the same what I told you about. Next. So this just every week this is a Google sheet which has to be filled. Every center fills up the data and gives the details, how much sequencing they have done and what clades and what variants they have identified. So the Government of India can monitor it very closely. Next. So the first case of COVID occurred in Rajasthan in March 2020, and the samples were sent to NIV Pune, where they sequenced it and they found it to be clade G, and the patient had come from Italy. From there on, we will see that the journey was to different mutations and different clades emerging. Next. So this is the clades G, which is there in our sample. Next. So then the new UK strain, we found alpha strain after a few months' time, and then not many cases of alpha strain happened in our, but we could, this monitoring was done by sending the samples to Delhi or to NIV Pune. Next. So similarly, this indicates what is, which strain is prevalent throughout the India. Next. So this is just indicating the amino acid changes which are occurring. Next. So this is indicating that mainly the G clade was present in Rajasthan. Like you can see, while the other parts of the country had different combinations of the variants prevalent in the different areas. Next. So many times we found out that Maharashtra was the first one to have many new variants, because a lot of people were coming from abroad and visiting there. So then after a few weeks time, the rest of the country followed suit. So we also found similar trends, and then these are the trends which were found over the period of time. Next. Next. So when we did the clinical profile at Rajasthan of these and matched them with the genomic sequencing data, we could see that the Delta patients, their profile of clinical symptoms were different than the Alpha 1 and the original G strain. So we could very clearly demonstrate the difference which was occurring in the clinical profile. Next. Next. So these are all the various waves. We have different variant emerging during the various waves in the India. I think it's not too clear from here. Next. So this is again another diagram. Here it shows that Rajasthan multiple variants were prevalent, while down south only a single variant was prevalent, 1.617.2, like the Delta had arrived other parts of the country earlier, and the Rajasthan we were having multiple strains. So we had to deal clinically, the strategy was decided accordingly. Next. So this is the picture of a lab. We have a ion torrent platform, and we are doing the sequencing, whole genome sequencing on this ion torrent platform, and using an automated Ion Chef system and also the manual OT2 platform to do the sequencing processing. And this is our team. Next. So the protocol we followed was the standard protocol of RNA extraction, then confirm the RT-PCR, and do the qubit and library preparation, and then template preparation, and put it into the chip for sequencing. So we could, initially 4 to 5 days it was taking us for sequencing, but we had batches of people. So on a daily basis, in the peak time of Delta and Omicron, we could actually do 200 to 400 sequencing each day, because we had a whole team working round the clock for doing this work. Next. So this is just shows how you have to do the quality check. You have to see the read lengths and how much loading was done on the chip. So that all you have to see to know what results you are going to get. Next. So this is part of that quality check. You have to see that they are 95% bases are aligned. So you know you're going to get good quality data, and your interpretation and analysis would be good. Next. So once doing this sequencing, we put the FASTA files in the pangolin software available free online, and that could identify the lineages. And those lineages, then we had to share the results initially with NIV, and then later on, after hand-holding, we could release them directly to the public health system. Next. So these are some of our publications during the process of Omicron and XBB 1.16 variants. Next. So during this process, we found out that end December 21, was the first case identified of Omicron, and within 5-6 weeks, it was 90% the strains were Omicron, and it spread really very fast. Of course, the mortality and morbidity was not very high, and the hospitalization also was not very was high, and this is one of our publications depicting that. Next. So these are the various variants studied over the period of time. We were getting samples from Rajasthan, and these are the variants. Next. Next. So in 21, September 21, Delta was the highest. 90% of the strains were Delta variant, while in March 22, it was 100% were Omicron. So by regular genomic surveillance, we could monitor the changing trends in the variant of the COVID, SARS-CoV-2, and alert the public health system. Next. So this is again there was a scare then sometime in March 23 that XBB 1.5 variant is coming from USA, many patients. So we had few of these cases, but then we followed up the patient. They recovered very uneventfully, and we did not have much of this variant in Rajasthan. Next. Next. So, but we did get a large number of, like within, again, 4 to 5 weeks time, our profile changed from Omicron to XBB 1.16, and large number of cases started emerging of this throughout the state. But again, here clinically, though the transmission was very fast, even in uninfected patients, the positivity was seen. And even the vaccinated cases, we saw the positivity, but only the comorbid patients, there was mortality in the patients with comorbidity. Next. So this is a study from Delhi, which again depicts the changing profile. Next. And this is the study from Maharashtra depicting how the XBB 1.16 variant emerged after March 23, February 23, and within a few weeks it was max 90% was XBB 1.16. Our data was also similar to that. And next. So this shows the comorbidity when they were present. There were only very few death cases, and they were more in the comorbidity cases, and the positivity was high. And this is the multiple mutations which were present, and the overlapping mutations, because there was also XBB 1.5 which was circulating, but it was the XBB 1.16 which was at the peak. Next. So the carry home time messages, we need to carry out regular surveillance, and initially the INSACOG kept changing the guidelines depending on the need of the day. Initially it was 5% of the samples to be tested, later it was 30 samples per 10 sentinel sites, and then eventually our state asked all the centers from the state to send samples, 5% samples, and when positivity was low, all the 100% samples were sent to us for sequencing. So depending on the workload and the need of the day, the state and the center kept changing the guidelines, and that helped us to identify the emerging variant and also control the spread of the disease. And so we have sequenced more than 15,000 isolate samples of COVID. And we are one of the high throughput labs for sequencing and for COVID testing. And we could help the state in the second and the third wave by quickly identifying the variant. So right now we positivity is very less, but the last variant which we were having were XBB 1.16, but there was no need to panic, not much of a mortality and morbidity.
Thank you,
there.
Professor Bharti. Yeah, but you have covered the journey, entire journey of first an emerging and then fading epidemic. And I think we thank you actually. And any people who would like to ask anything, I think better during the tea time, because you have done a very comprehensive coverage actually, which even citing the original papers from the state also. Thank you very much. Wonderful.
Thank you, sir.
Thank
Sorry
you
for
so
the
much.
glitches and for bearing with it.
Thank you, ma'am. Thank you, ma'am, for enlightening us on the genomic sequence surveillance of this COVID-19 virus. So next, I invite the next speaker, Dr. Mandira Verma, ma'am, from VP Chess Institute, New Delhi. Ma'am will brief us on the emergence of non-tubercular mycobacteria.
You can also change from here also. And you can change here.
Good morning, everyone, respected chairpersons, seniors and friends. And thank you, Professor Ramachaudhary, for inviting me here, and also thank you for including non-tuberculous mycobacteria in this session. NTM is something that's really close to every mycobacteriologist's heart. So I'll just move. Page down. So just to recapitulate, NTM, as we know, are ubiquitous, and that is why for several decades, they've just been considered to be contaminants. So they've found in soil, in natural water, in treated water. In fact, we found Mycobacterium fortuitum in some of our tap water samples in our own laboratory. So that is why they're so commonly, they contaminate samples. But they have now been known to be potential pathogens, and usually there's no person-to-person transmission, although there are some reports that are coming in now that patients of cystic fibrosis, there may be some person-to-person transmission, but usually there is none. And with advanced technology and so many sequencing facilities, there are more than 200 species that are now recognized in NTM itself. So just briefly, since when do we actually know about NTM? So it was in 1868 that tuberculosis in chickens was described and was found out to be due to Mycobacterium avium. It was nearly 80 years later that the first human case occurred, and this was in 1943. This was a patient who had silicotuberculosis in USA, and then they found out that this pathogen was actually Mycobacterium avium. And after that, of course, pulmonary NTM were described and recognized, and then with HIV, Mycobacterium avium became, of course, more common. So this takes us back to our college days when we used to read about classification and used to read about Runyan's classification. So 2 points about the slow growers and rapid growers, they're still used, even, there have been decades since I was in college, but we still use them as slow growers and rapid growers, slow growers taking more than 7 days to grow. But recently, 2018, there was this paper by Gupta et al., and they said that when we club the NTM together, we are actually clubbing together species that may not be truly related. So they said with so much of sequencing and so many other technological things available, we should actually be using insertions and deletions. So they used conserved insertions and deletions, and they used conserved signature proteins, and then they described Mycobacterium into 5 genera. So one was Mycobacterium, and there were 4 novel genera. One was Mycobacteroides, that included Mycobacterium abscessus complex, the Mycolicebacterium, which was Mycobacterium fortuitum, the Mycolicebacter, that is Mycobacterium terrae, and the Mycolicebacillus, which is trivial. So these are the 4 novel genera. However, this publication came out, but these generic species are still not validly used, so we continue to use the term Mycobacterium. Now just briefly, NTM, just like TB, also majorly cause pulmonary disease in 80 to 90% of the cases, but extrapulmonary diseases like lymphadenitis, skin and soft tissue infections, and bone infections are also known. And most of the samples that we get, although I'm from a chest diseases hospital, but we do get samples from outside. Most of our samples belong to people who have had post-surgical wound infections, and they turn out to be NTM. So we said that NTM were thought to be just contaminants, but now we know that they are potential pathogens. But who are the people who get these diseases? So there are certain predisposing conditions. Of course, one of them we all know is immunocompetent individuals. If they're on immunosuppressant therapy or HIV patients, etc., they will be predisposed to NTM infection. But even in immunocompetent individuals, it's been seen that there are certain predisposing factors. Like the lung should have a structural damage, for example, in bronchiectasis or cavitation. There are certain host conditions like elderly people, even women are more susceptible to NTM infection, or vitamin D deficiency, and certain polymorphisms. I don't think it's very clear here, but certain polymorphisms like cystic fibrosis regulator gene polymorphism, that also makes the patient susceptible. So then we come to why exactly this topic was selected for today's session. It is emerging, as is it really emerging, or is just being recognized? So yes, it is sort of emerging because, as I said, there's increased life expectancy, there's larger aging population, and they're more susceptible to NTM infection. And then there are climatic changes. Of course, we know that there are globally climatic changes taking place, which can change the flora. Plus, most importantly, microbiologists and physicians are aware that NTM are not just contaminants. Several years back, they said anything that was not rough, buff, tough, indicating M tuberculosis was usually just taken to be a contamination. But now we know that we have to look at it more carefully. And of course, there's so much of improvement in technology. We've just heard about artificial intelligence. We've heard Dr. Bharti's talk. So much of improvement in technology that we can now have easier methods of looking and speciating mycobacterial genus. So coming to global epidemiology, I just wanted to share this. It's because there's a plethora of information on NTM. But when we come to global epidemiology, this paper shows that there is a geographical variation. So if we see on the left side of the graph, the blue portions show Mycobacterium avium complex. There is a difference in the various continents. On the right side, the red portion is actually showing Mycobacterium avium. So if we just consider MAC is seen in Australia, but when we and South America and North America, but when we consider individual species, South America and North America have more of Mycobacterium avium, and Australia has more of intracellular. So there is a geographical variation which we should be considering. This is a beautiful review which came out in 2022, which shows exactly what you're talking about, that there is a changing trend of NTM. So as we can see, the red areas show that there is an increasing isolation of NTM. Even India, we can see there's an increasing isolation of NTM. When they say infection in this paper, they meant isolation. However, the correlation with disease is not that much yet. So we need more data on this. So when considering all that we had read, we wanted to see what is the condition in South Asia. So we wrote this review. We took papers from South Asia itself, and we found that majority of the papers that we included in our review, leaving out case reports, etc., majority were from India. There were just a few papers from the other countries. So we do need more data. But even in this, in the 17 studies that we included in our review, we saw that most of them, 50% of them, had not considered any of the guidelines to see if the organism was clinically relevant or not. This is another review that we wrote taking into account skin and soft tissue infections of RGM, rapidly growing mycobacteria. Again, most of the papers were from United States. There were quite a bit from Southeast Asia, especially Taiwan. There were a number of them from India. And the most common organism was Mycobacterium abscessus, followed by Mycobacterium fortuitum. So that was about epidemiology. Now we come to the diagnosis. So before I talk about microbiological diagnosis, just briefly, the clinical and microbiological criteria that are required to say that a particular isolate would be responsible for a disease. So there are criteria that are laid down that we should have at least 2 positive samples from sputum, or we should have 1 positive bronchial lavage or histopath and sputum or bronchoalveolar lavage. So a single isolate alone would not be responsible. And even when we look at 2 samples, it should be the same species. And in case of M. abscessus, it should be the same subspecies. It's not that we are isolating fortuitum in one and then next we are isolating abscessus. So that would not be the case. Then if we find a patient, if the physician finds a patient, they're suspecting NTM infection, but it's not falling into the diagnostic criteria. We should follow it up till we rule in or rule out that infection. Institution of therapy depends on the risks and benefits. So pure isolation, obviously, this is a potential pathogen, not an obligate pathogen. So one has to consider the risks and benefits. And if there are rare NTM found, as in my future, other slides, I'll show our own experience. So we do find new NTM, and one should consult experts for that. So then we come to the microbiological diagnosis. Of course, culture is irreplaceable, and both solid and liquid culture would improve the sensitivity tremendously. So both can be done. And of course, there are certain techniques which vary from when we do it for Mycobacterium tuberculosis or for NTM. So that has to be considered. The NaOH concentration should be lower. There may be different incubation time periods for slow growers and rapid growers and so on. Once the culture is done, we have to obviously identify. Now, earlier we used to do biochemical tests, and our reaction was like literally a lot of frustration, because with all those biochemical tests, we would still not arrive at the conclusion. But now, of course, with MIDGET and with MPT64 immunochromatographic tests, we can at least rule out NTM. We can differentiate between MTB and NTM really fast. So, as in the rest of the field of microbiology, there are lots of advances that have taken place in NTM identification. I'll first cover the ones which involve lipid and protein profile, and then the genotypic. So this is HPLC. Everyone knows about HPLC and the problems we face with HPLC. They're expensive instrumentation, and you have to compare the profiles with known profiles, known databases. But work has been done on HPLC, and this paper that I've quoted is from NTI Bangalore, and they found 95% identification of 10 different NTM species. Malditov, I think, has come up in such a big way, and but with experience with mycobacteria is limited. It is a complex preparation of samples. There are 2 models that are available, Vitek and Bruker. Bruker has a much wider database, about 178 species that are available on Bruker. But there are problems with Malditov because we need an active growth for protein extraction. And for rapid growers, it's better, but for slow growers, there's a problem. So as in this paper, they showed that there was 94.5% agreement between 16S ribosomal RNA and Malditov in case of RGM, rapid growers, but in case of slow growers, it was only 77%. This paper came out recently in 2022, and they analyzed data from several laboratories, and they found the same thing, that rapid growers, it was easier to do Malditov than slow growers. But in rapid growers and slow growers also, individually, if we consider species, there is a difference between different species. For example, for rapid growers, M. fortuitum gives better results than others. So then we come to the genotypic assays that are widely used in most of the laboratories to identify NTM. The first is line probe assay. We use that very frequently in our lab. It's the Haynes test, the genotype Mycobacterium CMAS that we are using. And the turnaround time is much, much better than biochemical tests. Recently, the genotype Mycobacterium CM Direct has also come out, which can be done directly from samples. For the rest, we need to use culture isolates. But CM Direct is there in the market, although it's not readily available to us yet. Next is PCR restriction enzyme analysis. That again is being used in every laboratory and very often. There are several genes like HSP65, RPOB, etc. that can be used, and several enzymes that can be used. One of the first papers that were published was by Talenti et al. They used it on HSP65. And another paper that is commonly quoted is by Wong et al. They also used HSP65, but they used different enzymes, SAO and CFO. So just quoting two papers that came out on PRA in India, and one was from NITRD. They used the Talenti technique, and this is our paper where we used Wong's technique, that is using SAO and CFO. The problem with this was that when you consider differences, some of the differences are too minute, and so we needed new-sieve agar, which is very expensive, to differentiate the bands. So we wanted a technique which would be cheaper and which would be faster. So in our lab, these are my students, Garima and Anshika. They've of course got their PhD now, but they worked on this new technique that we devised, where we used HSP65, but we used the enzymes NRU1 and BAMH1. So if we found that there was digestion by both, it was MTB, and if it was only by NRU, then or one or the other, then it was NTM, and if there was no digestion at all, then it was a non-mycobacterial species. But again, we did use an enzyme. We wanted to get rid of that, so we devised another methodology, and my student Kamal worked on that. So here we just used simple PCR for two genes, HSP65 and RV145HC, and if we got digestion, if we got both amplicons, then it was Mycobacterium tuberculosis. If we just got a PCR amplicon for HSP65, it was NTM. And if there was no amplification, it was non-mycobacterial. So we use this technique every day in our lab because it's so simple to use. And this is what we got from over the past few years. These are the NTM that we've isolated. The left-hand columns show the non-clinically relevant, which are much more than the clinically relevant. So obviously for the clinically relevant, we considered all the criteria that I've just discussed. So then I'm just interspersing our experience with the technology that I'm discussing. So then we come to genomic sequencing. Genomic sequencing also, everyone is using it. We use 16S ribosomal RNA, but of course it is not as good as we thought it would be. So HSP65, RPOB and ITS regions are also being used. HSP65 for slow growers and RPOB for rapidly growing species. Whole genome sequencing, now it's become so cheap now. A few years back it was about 70,000 per sample. Now it's come down to even 8,000 per sample. And whole genome sequencing can give all kind of data, not just identification, resistance, genotype. So now our experience with these two techniques about gene sequencing and whole genome sequencing, this was an experiment that we did on mouthwash samples. I won't discuss that because there's a shortage of time, but we just wanted to see if there really was transient flora of NTM, and we saw that there was one unidentified organism. That organism could not be identified by the CMAS. So we did sequencing. We sequenced it using the ITS region. It came out to be Aropensi. Then some sequencing gave us Heracleonense, all M-terre complex, but different species. And after working on it for several months, we used different databases, we used different sequences, and finally we used the digital DNA-DNA hybridization, which an expert told us was the gold standard actually, and we were told that it's a new species. So this new species, we've tentatively described it as Mycobacterium delhi, and we described it biochemically. It's a slow grower, and we performed the MIC for the drug susceptibility test. So it was susceptible to the commonly used antibiotics, but of course this is from a normal mouthwash. It's a common normal flora. We're just describing this as a novel species to highlight what I said earlier, that there are species that are emerging. Then the drug susceptibility tests. So this is the last few slides of mine. So the drug susceptibility test, CLSI says we should use broth microdilution technique, and that for MAC, cancer C and abscesses, it's better to do the drug susceptibility instead of empirical therapy. And for abscesses, also macrolides should be tested. For the incubation period, even though it's in a rapid growth, should be 14 days to rule out inducible macrolide resistance, or we can look for resistance to mutations in the ARM gene. So here the review that we had written earlier, we also studied all the drug susceptibility profiles that people used, and different techniques were used. Some people used E-test, some broth microdilution, and some even used Kirby-Bors. So just to highlight that we should have a standardized technique so that only one particular technique is used. This was just quickly our data on the RGM that we had isolated. So they were mostly sensitive to amikacin, clarithromycin, and linezolid. Of course, M428 was, so they were susceptible to amica, clarithromycin, and linezolid, and M428 was resistant to clarithromycin. This is the last case that I would like to share. This is a patient of actually, he had COPD, and he came several times to our lab. The first time we isolated Mycobacterium abscessus, it was sensitive to clarithromycin, and the patient was treated, he got well, but next year he came again with Mycobacterium abscessus, and then again in 2022, Mycobacterium abscessus. In 2023, there was no growth. So earlier we used CMAS, and we identified it as Mycobacterium abscessus. We did the MIC, and it turned out to be susceptible, and the patient was well for intervals and then not well, which is very well known in Mycobacterium abscessus. Now recently we got the NTM-DR strip. So this is another way to look for drug susceptibility tests. The NTM-DR strip looks for not only resistance to macrolides and aminoglycosides, it also, we can make out if the Mycobacterium species, what subspecies it is. And we had our isolates, when we tested our isolates with the NTM-DR, we found that the first isolate actually, when we got it, turned out to be Mycobacterium maziliense. The last two isolates that we got were Mycobacterium abscessus subspecies abscessus. So actually, if we looked at the clinical diagnostic criteria, we should not have even considered Mycobacterium abscessus. So there are these issues that are, that's the dilemma that a person working on NTM and a person treating NTM in the clinic faces. So just the summary. There are challenges for NTM. There are several unmet needs. The clinical microbiological aspects and criteria need to be worked out. There's several new species, as I just showed from our example, several new species that are coming out, but clinical relevance needs to be worked out too. We have several techniques. We have 16S RNA sequencing. We have whole genome sequencing and lots of other techniques. But as one of the speakers in the students' symposium said, that we should use, make excellent use of technology than use excellent technology. So here it really fits in because there is so much of dilemma with NTM, but a lot needs to be done on NTM and to find out, because they are emerging and they are definitely causing disease. So with this, I'll end my talk, and these are our contributors for the data that we shared. And thank you.
Thank you, Dr. Mandira. Thank you very much. Very comprehensive presentation, including what you are doing. I just one quick query. Any Rajasthan specific data in this actually?
Sorry, sir?
Rajasthan specific data from Rajasthan institutions?
So I think there was one data from an institute, but we could not include it in our review because that was a case report.
Yeah.
So we just included the reports that were that had original
No,
the original
that's okay. I
articles.
understand that, but over the 50 years, a lot of the case reports have come, which have
Yes,
their
sir.
own meaning, actually.
Yes, sir. Yes, sir.
Because others are not. But I must inform the listeners today, I think Mandira will be part of those projects. There are 5 projects going on at the moment, 3 funded by DBT and 2 by ICMR, in which we are looking at, what should be the algorithm for India. Because the
Right.
criteria for India will be different. What we will recommend for the nation, like, what Bharti was talking for how you did the surveillance for Rajasthan, for example, that way we will have our own guidelines for the India.
Right, sir.
And I think by the 2024 or 2025, we'll have the sufficient data to come out with the Indian specific guidelines on NTM. So I think watch for them, listen to those people, and Mandira will maybe part of the 1 or 2 projects out of those 5, because those are the projects which are from our people from Delhi. I think 2-3 labs are involved, then Northeast, everywhere their involvement is there. I think let's look with the optimism and with a lot of analysis of the Indian data, what you look at, how India is different than the others.
Right, sir.
But congratulations to you for your very, very excellent presentation. Yes.
Thank you, sir. Thank you.
Okay. So train more people from these places also who come to your lab, host them, so they also learn and start doing the things here actually. Yes.
It
Good.
will be a pleasure, sir. Thank you. sir.
Okay, okay.
Thank you, ma'am. So next I invite our next speaker, Dr. Amita Joshi, ma'am. Ma'am is a professor and Department of Microbiology, Grant Medical College, Alibag, Mumbai. Ma'am will brief us on emergence of XDR tuberculosis. Welcome you, ma'am.
Where do I work with?
Yeah, it's working. You just page down.
No. Okay, the first slide went off. So it's good afternoon, everybody. I thank Dr. Rama and Dr. Shama for giving me this opportunity to speak on emergence of XDR. And I think the glitches are there, no issue. If you look at the India TB report that was presented by our Honorable Health Minister, they said, and this is post-COVID, that in 2021, we had an increase by 19% of TB cases in India. The total number of incident cases also was higher as that seen in the first few months of 2020. The deaths also were higher by nearly 13% post-COVID, which we saw. And the prevalence of all forms of TB was the highest in Delhi and the lowest, which was seen in Gujarat. Looking at the resistant, or what we call the MDR-RR, as per the global report, we have more than 1 lakh cases in India itself. And if you look at our own NDRS survey that was done, the first of its kind in the world, and it was done in India, we saw that nearly 28% of TB cases were resistant to any of the drugs that is there. The definition of MDR-TB and XDR-TB has gone a little modification over the years. MDR-TB, as we all know, is that which is resistance to isoniazid and rifampicin, with or without resistance to other drugs that are used for the treatment, as well as what we were calling as pre-XDR, that is the quinolones also, has been taken into the MDR. And XDR, which was initially in 2006 said to be that MDR plus quinolones plus injectables, have now been modified. As you are aware, that injectables are literally out from our therapy, and therefore now when we say it's an XDR-TB, it means an MDR plus quinolone, and here we are only interested in the levoflox and the moxi, which is part of my therapy, and at least one of the additional group A drugs, that is bedaquiline and or linezolid, that is there. Coming back to the DRS survey, like I said, it was one of the kind in the world which was done, a very systematic survey which was done. The MDR was around 6.19%, pre-XDR. Again, this is prior to the ones of the newer definition which is came over here. And the reason I'm harping about the XDR and what definition I'm doing it, because if you ask me in true sense today, is it emerging XDR? I will not be able to tell because we don't have diagnostic or the laboratory proof to that effect. Why? And I will mention as I go through my talk itself. And if you look at the studies from across India, which was again speaking prior, that is the older definition, it ranged anywhere from 0.3% to as high as 60% across the scenario, okay? If you look at the factors for XDR-TB, it is more or less similar to your MDR, your pre-XDR factors which is there, and that main stresses upon either a faulty or improper therapy that you have initiated, non-adherence to the treatment guidelines, incomplete or inappropriate or erratic use of the second-line drugs. As per one study which was done by South Korea, it also gave, there was inaccessibility to labs which gave a reliable lab report for the DSTs. And as per one of the Indian studies, it said that it is the lower socioeconomic, which is our issue over here, as well as comorbid illnesses and previous intakes of the second line, which were responsible for XDR. The diagnosis of XDR is just not possible clinically. However, a clinician can only suspect XDR if there is a failure to an MDR therapy and the person concerned is a close contact who has already received therapy of XDR-TB. The only way to diagnose XDR currently is by doing what we call as drug resistance or drug sensitivity testing, first to the first line drugs to say it's an MDR, and of course then go ahead and say that it is also resistance to the second line drugs. Coming back to my topic as to what XDR that we talk about, if you look at my, or rather the Government of India treatment guidelines, it's an all oral regime, whether it is for sensitive, where we use a 6 month, may be extended in few cases, have been shown over here. And the drug resistance TB also, which is over there, is based upon what we are giving certain, what we call as UDST. That means upfront I'm doing a NAT, and then going on to do the second line DSTs, followed by liquid DSTs for some. And we have what is known as the H-mono, H-poly for isoniazide resistance pure. And of course, the shorter or the longer BDQ based upon what results you're going to get on the first line, that is your isoniazide, whether low level or high level, and what you get from the quinolone. So the drugs, if you go to see, you'll ask me why I'm teaching you therapy, because the lab has to go hand in hand with what is the drugs which is available to make a regime, because then only you will be able to give justice to what we say as a lab diagnosis of XDR. So group A has, and ideally a clinician wants all these 3 drugs to be part of his regime, that is a quinolone, a Bidacue, as well as the linezolid. He may add 1 or 2 of group B, which is your clofazimine, where sensitivity or DRTB testing is a big challenge, and of course your cycloserine may be there. And all other drugs that we have to make a regime belong to what is known as the group C drug, which is there. So if you look at the technologies which are available to diagnose, say XDR-TB over here, and what is available, the one in dark blue is what is available. So whether my NAT or even the XDR, what we were talking about, or my second line LPA, which as per the 2006 definition was giving me injectables, but as per the newer definition, it does not give me the diagnosis of an XDR-TB. So what do we have which is endorsed by WHO and available but not in my programmatic mode is your target sequencing and your sequencing. I'll go into the pros and cons of that also. So based upon the ease of which we does, we have on the lower rung, and that is what we have put up front in a diagnostic algorithm, is the NATs that we have. And then based upon the complexity, we have upper and upper, which is going, your LPAs will coming, and then of course your sequencing, which is right on top of this pyramid, which is over there. The methods which are there in my National TB Elimination Program, out of the various methods which are there, 2 only, as per the older definition, were there for XDR-TB. Though, if you look over here, expert XDR is mentioned over here, 2019, but it never came into the program for various policy decision which is over there. But this is the only molecular test which was available as per the older definition, which also is able to give me a diagnosis at least of pre, because quinolones are addressed, and your second line LPA. We have to rely on the cultures, and we'll also look into that. And based on this, looks very complicated, but simplest is that whatever sample upfront you give them a NAT, whether you're giving a CB NAT or you're giving a true NAT, and based upon the findings of that, if there is resistance not detected, think or rule out at least isoniazide by doing first line LPA, and if it is resistance to isoniazide, we start what is known as the mono or the poly, the drug therapy for the treatment, and if it is not detected, of course, the drug sensitivity therapy goes on. On the other hand, if it is resistance detected, the upfront first line, second line LPA based upon the INH, whether it is INHA or CAT-G, that means one on the two only and no quinolone, we start the shorter or the BDQ regime, whereas if both, that is INHA and CAT-G, with or without the quinolones or only the quinolones also, we go in for the longer therapy. So the clinician is relying on us, that is the microbiologist, to give a guidance as to whether what therapy he is going to start for this. So like coming back to the two tests, which I'll go a little bit in detail, because by the previous definition, that was what is in use, and presently in our this we are using, yes, I'm talking about XDR, though it has not come, like I said, in my diagnostic algorithm. But it is available in the market. There are laboratories in the private sector and many in the government sector also, though not in the program mode, which is using this XDR cartridge, which is called, okay. If you look at the targets that are over there, for isoniazide, there are two additional targets which we don't see in the LPA first line. Otherwise, all the other targets are more or less similar that I'm going to see in my first and second line combined LPA. You will ask me why isoniazide. Even we don't know why isoniazide came in the XDR, because that is my first line drug which I'm using for therapy. Advantage, definitely, if you compare it with my LPA, because that's the only molecular platform. It is rapid, it is automated, it doesn't require, you already have a CBNAT setup, where you may have an AC room, you may having a UPS, which help. And the main thing is the turnaround time is around 2 hours. Like I said, many labs across India are already using this XDR module. And the main advantage above the LPAs is the higher sensitivity to detect MTB, faster time to response, simple handling, and number of invalid tests, which we see especially with second line LPA, especially if it's smeared or low positive smear sample, then we do get invalid results in the LPA. The limitations, except for XDR, you need the 10 color module. You cannot do with the older machines which are there. Of course, there is a software upgradation if offered by the company very well, otherwise you have to buy the machine. The other limitations of machines are similar to that of my CBNAT machine. Coming to the assess process itself, remember a negative test does not rule out the X-ray, and the reason being the limit of detection of this XDR cartridge is quite high, up to 136. So it does not rule out, because if the load is less, I will not be only detecting. Similarly, you can only say resistance detected, but you cannot say it is susceptible. So you can rule in resistance, but you cannot assume that there is no resistance, that this individual is susceptible, okay? Because the targets are very specific over here. And for ethionamide, it has used only one, that is INHA. We know there are other pathways, ETH-A, ETH-B, many more pathways which are there, but the INHA reflexly which we use for the low dose resistance in isoniazide is the pathway also similar, and therefore we can say that ethionamide, which is the third or group C drug, cannot be used to make the regime for these individuals. Also, there are additional, I said, 2 more targets which are there in the isoniazide, but evidence has shown that if you do not get in INHA and CAT-G, but you get in these 2 targets, you have to be a little careful, because there is not much that it can be conclusively said that it is resistance to isoniazide. Then when you get indeterminate results, it may be because the limitation, like I said, targets, the targets are not there, so it may be some rare or new. You cannot just rule out as a machine error. You may have to repeat those particular tests. Mixed population, which is very common in TB patient, which it occurs and you're using an XDR cartridge over there, and especially if the resistant population is low, then you will not be able to detect with this, what you say, XDR cartridges. Also, when the low count only in the bacteria is there, it is not because the limit of detection. We are using in the national program for MDR, we are using a generation 4, which has a limit of detection of 113 bacilli or CFU per ml, and the ultra, which we don't use in the program, but which is available across India, has a limit of detection of which is as low as 13. So supposing your first test is done by either your generation 4 or by your ultra, and then you happen to do, especially if you do an ultra and happen to do XDR, you won't also detect because it's a very high, that is up to 136. You can make the limit of detection down provided you concentrate, and it comes down to 86, which also is very high as compared to the ultra cartridge that you're using. Coming to the second line LPA, which we are using from so many years in the program and across India, which is over there. So the requirement remains the same for the first line LPA, which is there. And because there is a manual interpretation or manual this, you may be, and of course the 3 setup rooms which you do, we have to take care that we do not have controls, or rather we don't have contamination between while processing the sample. The main I'm stressing over here is only looking how we interpret the quinolones over here, because that helps the clinician whether he can give the quinolones or he cannot give the quinolones, okay? And that is by looking at the mutation pad. Fortunately for the clinician, the NICSHA, if your entry is there and you're looking at the results in the NICSHA, this guidance is given in the drop-down menu. The minute the lab reports the banding pattern, the clinician gets its, what you say, relation over the clinical interpretation of that report. So based on that, ethionamide, if I have got only CAT-G, I can think of taking ethionamide, which is my group C for the treatment, whereas if I have got the quinolones, which is over there, if I have mutation band 1, that is mut 1, mut 2 or 3A, I know I can yet give quinolone, that is a high dose moxie, but if I get a mut band that is 3B, 3C, 3D, then even quinolone is out from the therapy, okay? So advantage, like I said, can be performed directly from the sample, okay? You don't need to make a smear, but especially for the SLLPA, they say directly from the sample. Reports in 5 hours when I'm saying, meaning you have already done the first line LPA. So it is one additional day as after your first line LPA. Allows appropriate, that is what a clinician wants, how fast I can start the therapy in this individual, okay? And therefore the faster you start, the reduce the transmission and the spread of MDR-TB. The limitation in paucibacillary, whether it is in HIV patient, whether it is in the pediatric, whether it is in extrapulmonary, of course you can't do it with extrapulmonary sample. The second which I said is the contamination, which is very high. Reports are not as fast as that you get in the expert XDR, and you need infrastructure dedicated for a PCR setup, which most of the labs across. Post-COVID, 100% the labs across India have got an MCI making it compulsory, even all ones which are giving PGs need to have a PCR setup. It's a low through output unless if you have got a GT plot where you can increase your output to at least 40 per cycle, and inability to indicate all the steps, which is for any target sequencing, which is over there. So if you look at the accuracy between the expert ultra and the second line LPA, it's more or less similar. I've put purposely version 1, version 2. Version 2 is the one which we are using across in the program. And for quinolones, only look at the quinolones, the sensitivity, specificity is more or less same. So the only plus point is it has less, what you say, it's a closed system, faster results, which is giving the expert XDR an upper edge as compared to the second line LPA. Any rapid test which you are using molecular, whether I'm talking about NAT, whatever may be the NAT or this is there, definitely has reduced my turnaround time, which may be as early as this. When I want to do the MDR, that is up to 2 hours. And if I use the XDR, another 2 hours, which is over there. But it is not doing anything to the drugs which I am presently using for treating a second line, and that is your group A drug. It is not giving me anything for my bedaquiline. It is not giving me anything for linezolid. And therefore, like I said, only quinolones are being addressed at present, okay? Also, these stats are only covering, which is also there for the other, if you take target sequencing, only covering the targets which is commonly associated with resistance. Therefore, there is a gap which exists in these NATs which are being used. So, can I rely on the culture? Solid cultures, we are more or less given because of the long turnaround time. We only use them as backup, or like ma'am said in the previous, we are using it for NTMs, which is over there. Otherwise, we are relying on the liquid cultures, and that also the midget 960, that which we are doing over here. In 2018, WHO had come with a critical concentration. Ideally, the footliner in this also stated by WHO, you need to have your own critical concentration for your own country per se. But we are using the critical concentration which was done, and when you have a little doubt, then the particular lab does it. MIC is not in the program mode, which is over there. Only two I have highlighted where I get good evidence with molecular as well as the phenotypic, and that is again my quinolone as well as my injectables, with I'm not getting anything for the BDQ, linezolid, or other drugs that are used for the therapy. Liquid culture, yes, it is faster as compared to your solid cultures, but there also it may be as early as 4 days, but you have to wait for 40 days, for that is the cycle of my midget. Then you start the DST, which itself takes 2 weeks for most drugs, except pyrazinamide, which takes 3 weeks. Phenotypic DSTs, which we say is the gold standard, is inaccurate and not reproducible for pyrazinamide, ethambutol, clofazimine, ethanamide. I said I will be stressing, and these are my drugs which are forming my regime in the case of the MDR. Okay, so this is as per literature, not to blame the lab concern. There are issues with this. It is not available for BDQ, not available for delaminid, not available for pretonamide, and these, especially the first two, are part of my regime. There are few labs only in India which have got the powder of BDQ, and I think only one lab has got even for delaminid. Otherwise, once you have the powder also, which is not in the program mode, but we have to take it from, as a research, it is given by NIH and then pass the PT. Few labs are there in Mumbai, our lab in JJ as well as Hinduja lab have, and we are giving this service of phenotypic DSTs for only BDQ, not delaminid, because the powder is accessibility also is not there. Then we do not have agreed DST methods which have been established for some of the drugs which are used in the therapy. So coming back to square one, what do I do to first say that this is a case of XDR? So here comes, what do I do? And also liquid culture, if you rely, I can't tell it is mixed infection, which is common with TB patient, or heteroresistance, okay? So these two are not going to be taken care of with your liquid DST. So then comes, can we go in for, now I'm moving a little bit away from the program mode and asking whether I can use these two, that is a target sequencing.
Amita, 2 minutes.
Yes, I'm just summing it up over here. And what we have is to use these targets, either you have your own test, or we know that if you use the targeted sequencing, the main plus point of targeting, it talks about working directly on the sputum, and we have used this test Diplex, which is over there, and shown that it can be used. This is the kit which shows to most of the drugs which I'm using at present, 13 drugs which is giving a very nice report, telling you about mixed infections, telling you about the thing. The only thing is, if it is less than 2 plus positive, that means 2 plus and above it does, but not for smear negative, or 1 plus if we cannot do it over here. We had shown this finding and shown its utility for giving. 3 more kits were also done by WHO and published in 2023, which again, Diplex took the upper hand because of the more targets and also because it talked about lineages. For WGS, at present, if I go to see, it's only the culture, and that itself has an issues, especially because with sequencing, which is you need a concentration, so you should be certain what will be the technique that you'll use to extract the DNA. And out of all the platforms, 2 platforms only have shown promise, that is the MiSeq and the Oxford, that is the nanopore. The MiSeq, though it gives of short read length, number of errors are less as compared to your nanopore. The length, that is the read length is big, but the error rate is high. All these have to be correlated, so you have to rely on the catalogs, which is either the WHO or our own ICMR catalog, that when you get a mutation, is it actually associated? We published this because we did this in 2016, 2018, when the BDQ was introduced and clinicians wanted to know whether there is an underlying BDQ resistance. Fortunately, this was from the Govandi district, which was one of the site receiving the CAP, that is a conditional access, zero baseline DST, which we showed, or rather resistance to BDQ. The current use is to predict, identify strain. This is what the national program is using, and to dissolve, if at all, discordant results, to diagnose. Main is for basically for what we call epidemiology. And the main challenge is the high cost, interpretation, having technical, this is there. Just to summarize, the results of the various and the rates, if I have to do it over there, high rate for target sequencing, less as compared for the whole genome sequencing, not part of my program, but in the program, this is the rates in the private sector because all modalities are available over there. To control, I should basically control emergence. That means treat properly my first line, don't allow MDR or XDR to occur, diagnose it properly, use the test if you're using higher platforms, do vis-a-vis the phenotypic platforms. We know we are missing out, or we are rather only identifying 1 in 3 of my bacteriological diagnosis. 1 in 5 are getting actually the molecular, and only 1 in 3 of those who have been diagnosed are actually put down on therapy. So this is what has to be strengthened if I want to prevent XDR. Thank you.
Thank you, Professor Amitabh. Excellent presentation. But again, I think you have given a very clear message for everything what microbiologist can do at the moment to help the clinician.
Yeah.
But as far as the whole genome sequencing is concerned, the target gene sequencing is concerned, for the quinolones and for number of other drugs, they hardly reach 60 to 70% concordance,
Absolutely.
So that people must remember at the moment,
Absolutely.
the genome sequencing is not the answer for...
No.
Yes.
Therefore, it is definitely not for diagnosing.
Yes.
We are building up data.
Yes.
How much weightage will I give for mutations?
Yes.
For the Indian scenario.
Yes.
And though you are getting these sequencing results even in
Yes.
3,000 in Mumbai,
Yes.
we always tell a clinician, no, don't
Yes.
do so
Yes.
till we have data vis-a-vis.
you.
So
Thank-
that is one message I think I will put.
Thank you, Professor Amitabh.
Yeah.
Wonderful.
Yeah.
I'll request Professor Singh to offer his comments.
I congratulate all the 3 speakers. They have presented very well. Dr. Bharti, she has taken us through the journey of frightening COVID. Just not to remember it again, but we have learnt a lot lessons, and the technologies which have been made available to the farthest of the places in the country, and making use of these technologies in the diagnosis of the diseases. So with the COVID, we can do away with the diagnosis of other infectious diseases. Dr. Amita and Dr. Mandira, they have also presented very well, reminding us what we say, the tuberculosis, which is the king of the diseases, the death king. So, but still we are fighting with it, and we hope someday we are getting it eliminated, and recognizing the importance of non-tuberculous mycobacteria, which we earlier used to have discarding them as contaminants or not disease-causing organisms. But with the changing patterns of climates and the host-parasite relationships, more and more such organisms are being recognized, and we have to deal with them also. Then only we can get over with the tuberculosis from the human race. Thank you very much again, all these speakers.
Thank you all respected chairperson and speakers. So now I request the chairpersons to kindly felicitate our speakers by presenting a memento. So I request Dr. V.M. Kato sir and respected Singh sir to present a memento to our first speaker, Dr. Bharti Malhotra ma'am. I also request Dr. Rama Chaudhary ma'am to felicitate our chairperson, Dr. V.M. Kato. Thank you all respected chairpersons. So as we have just completed the first half of the first session, so now we will have a tea break of for 15 minutes, and simultaneously we will have the poster session one there along with the high tea. So we request you all for the high tea.